You are about to step into a mystery where science meets the unknown. Imagine an old family house at night. The rooms are quiet. The photographs are black and white. And yet there's a feeling that the walls remember. A grandfather who never spoke of the war. A grandmother who rationed food long after the famine was over. Their children grew up in those rooms, and their children after them. And sometimes the shadows in the house seem to travel faster than the stories. What if biology keeps a ledger of experience, not only in the brain synapses, but in the chemical markings that ride along with DNA? What if stress and safety, hunger and care leave notes in the margins of our genome? Notes that can be read by the next generation cells? This is the promise and the puzzle of epigenetics, and it leads to a question that feels almost mythic. Can trauma echo across generations? And if so, how far does the echo travel? And if you'd like to help me bring more of these mysteries to life, don't forget to like, comment, and subscribe. Tonight we'll walk from bombed cities and empty cupboards to mouse nurseries and molecular machines, and we'll ask what the science really shows, what it can't yet prove, and what it means for how we understand responsibility, resilience, and repair. Epigenetics began as a bridge between genes and form. In the mid-20th century, biologists realized that having the same DNA in every cell doesn't explain why a neuron and a liver cell look and act so different. Something must steer which genes switch on and off in each context. That something turned out to be a set of chemical marks and packaging choices, epigenetic marks, that don't change the letters of DNA but do influence how those letters are read. You can picture the genome as a library and epigenetics as millions of sticky notes and bookmarks. Open here, skip this chapter, read this page in a whisper. The best studied mark is DNA methylation, small molecules that latch onto cytosine bases, often at CPG sites, and tend to dampen nearby gene activity. Histone modifications, acetyl, methyl, and other tags on the protein spools around which DNA is wrapped change how tightly the chromatin is wound and whether the transcriptional machinery can reach the script. Add to this a chorus of non-coding RNAs, tiny strands that shuttle messages or silence them, and a 3D folding that brings distant pages together, and you have a dynamic score that tells cells what kind of cell to be, how to react, and when to remember. The revelation that experience can tweak this score arrived through animals long before it was whispered about in human families. In one famous experiment, rat mothers who licked and groomed their pups more frequently had offspring with calmer stress responses. Cross-fostering, swapping pups between high-lick and low-lick mothers, swapped outcomes too. And scientists trace part of the effect to epigenetic changes in genes that regulate the stress axis in the brain. In another line of work, mice carrying a particular agouti gene variant could be nudged by the mother's diet during pregnancy toward different coat colors and metabolic fates. The diet shifted methylation marks near the gene, changing its activity without altering its sequence. Elsewhere, paternal effects surfaced. Male mice fed high-fat diets, exposed to stress, or trained to fear a specific odor sometimes sired offspring with altered metabolism or altered sensitivity to that odor. Remarkably, injecting small RNAs extracted from those fathers' sperm into healthy zygotes could recapitulate some of the changes in the next generation, pointing to RNA, cargo as one vehicle for memories of the father's environment. These animal studies illuminate a bold possibility, that life events leave a molecular trace that can transit through egg or sperm. But mammals carry a built-in skeptic, a pair of reprogramming events that wipe most epigenetic marks clean. The first occurs in the cells that will become sperm or eggs. The second happens just after fertilization. This double rinse resets the embryo, turning the epigenetic page back to blank so a new body can be written. Some regions resist the wash, imprinted genes, which by design keep a parent of origin mark. Some transpose on rich stretches that are silenced for genome safety. And, as experiments suggest, certain small RNAs that can instruct new methylation or gene regulation in the embryo. The existence of these escape hatches means transgenerational epigenetic inheritance is biologically plausible in mammals. But it also means it is likely to be selective, fragile, and far from the simple idea that trauma marks your DNA and your grandchildren inherit those marks wholesale. When we step from mice to people, the terrain becomes messier and more human. History has provided natural experiments that no ethics board would approve. In a brutal winter near the end of World War II, a blockade starved a European city. Children conceived and carried during that famine grew up under circumstances that allowed careful follow-up. Decades later, some of them showed higher rates of metabolic disease. In certain cohorts, subtle differences appeared in DNA methylation near growth-related genes, among those exposed in utero to severe hunger. Another line of research tracked families from a small Swedish region through crop cycles and births. Grandfathers who experienced feast or famine as boys seemed, in some analyses, to have grandsons with altered risks of diabetes and mortality. Elsewhere, researchers studying descendants of genocide survivors reported differences in epigenetic marks in stress-related genes compared to matched controls. These findings, combined with reports linking maternal smoking or stress to molecular changes in children, fueled a compelling narrative. The body keeps the score, and sometimes it hands that score forward. But compelling is not the same as conclusive. Human studies face hurdles that even the most careful design cannot entirely clear. Most epigenetic data come from blood, a moving mosaic of cell types whose proportions shift with age, infection, and stress, and marks vary enormously between tissues. The brain, where many stress effects matter, is difficult to sample. People who experience famine, war, or persecution also experience poverty, displacement, discrimination, changes in diet and air quality, and altered access to care. These socioeconomic and cultural legacies shape biology through many channels: prenatal nutrition and stress hormones, parenting styles and neighborhood safety, infections and sleep, exercise and education. The epigenome is responsive to all of them, teasing out what portion of a small methylation difference in peripheral blood is inherited memory versus ongoing environment requires large samples, replication across cohorts, and careful control for cell type, ancestry, and life experience. Some early human studies have struggled to replicate. Effect sizes are often small. Plausible alternative explanations remain on the table. So where does that leave the idea that trauma can echo? A careful synthesis distinguishes between intergenerational and transgenerational effects. When a pregnant person experiences famine or stress, 3 generations are present: the parent, the fetus, and the germ cells forming inside the fetus that will one day become grandchildren. An effect on the child, F1, is intergenerational. An effect on the grandchild, F2, may still be intergenerational if it arose from direct exposure of those embryonic germ cells. In mammals, the bar for true transgenerational inheritance is F3 through the maternal line and F2 through the paternal line, generations not directly exposed in utero. Human evidence meeting that strict bar is suggestive, but not decisive. By contrast, intergenerational pathways are well supported. Stress and nutrition alter the placenta, the gatekeeper between parent and fetus. They shape hormones and immune signals in pregnancy. They influence birth weight and organ development, laying foundations that later life will build on. Paternal pathways exist too. Sperm carry not just DNA, but a cargo of RNAs and remnants of histone marks. These respond to diet, stress, and age, and in animal models they can alter offspring physiology. This is where an easy slogan, "You inherit trauma," gives way to a richer, harder story. Biology writes in pencil, "The epigenome is a palimpsest, always being revised by experience. Some pencil marks are darker and longer lasting. Others fade quickly. Early life seems to matter disproportionately because it sets baselines in endocrine and immune systems and shapes brain circuits during sensitive windows. But plasticity is the other half of the equation. In animals, enriched environments, stable caregiving, and exercise can remodel epigenetic marks and behavior, even reversing some stress imprints across development. In people, interventions that reduce maternal stress, improve nutrition, and strengthen social support have measurable effects on birth outcomes and child development. You can think of it like a soundboard. Adversity turns some dials up. Care and safety can turn them back down. The music's never exactly the same, but it is never stuck. If we widen the lens to theories, several candidates try to explain how echoes might carry. One proposes small RNAs as couriers. Stress in the parent alters these molecules in sperm or egg, and they, in turn, tweak early embryonic gene expression. Another emphasizes leftover histone marks or DNA methylation at sites that escape reprogramming, including imprinted regions and sequences near transposons. These could bias gene activity for a generation or two. A third points to the placenta. Because it is partly fetal and exquisitely sensitive to the parent's physiology and environment, it can retune nutrient delivery and hormone exposure with long-lasting effects. And a fourth, often underappreciated, is behavioral and cultural inheritance. Trauma changes how a family copes, communicates, disciplines, and soothes. Those patterns, passed by example and expectation, alter a child's stress landscape, and through it, their biology. None of these paths exclude the others. In the real world, they braid. Speculative ideas orbit the core. Some envision a kind of adaptive tuning. If a parent's environment screams famine, epigenetic marks prepare offspring to store calories. If the environment screams danger, marks prime faster stress responses. This could be beneficial when the child's world matches the parent's. The problem is mismatch. A body tuned for scarcity meeting abundance. A stress-primed brain in a safe home. Others flirt with neo-Lamarckian rhetoric. The experience of the organism writes back to the genome for the species. That's a bridge too far. Epigenetic inheritance, where it exists, is a local whisper, not a grand rewrite of evolution. Natural selection still shapes genomes over generations. Epigenetics shapes how genomes are read within and between 1 or 2 generations, occasionally more, with diminishing echoes. Why does this matter beyond curiosity? Because it reframes responsibility without fatalism. If experience can reach into the germline suitcase, even a little, then social policies that reduce violence, hunger, discrimination, and pollution are not just about present compassion. They are about biological futures. If safety and opportunity can soften hard marks, then investment in early life pays compound interest. It also shifts how we talk about blame. People carry histories they did not choose, but those histories are not destiny. The same plasticity that makes us vulnerable makes us malleable. The question, what happened to you, can be taken seriously without surrendering to determinism. There is also a humbling scientific lesson. The human body is not a hard drive that stores bits precisely, nor a clay tablet that preserves impressions forever. It is a living manuscript, annotated by life. We now have tools, single-cell sequencing, long-read methylation maps, chromatin assays, that let us watch the scribes at work. We can see how stress hormones nudge transcription factors that call in or dismiss chromatin remodelers, how inflammation leaves traces in hematopoietic stem cells, how sleep, diet, and exercise shift the epigenetic clocks that correlate with aging. We can even, in laboratory contexts, edit methylation and histone marks at specific genes to test causality. But we must be careful what stories we tell. Overselling epigenetics has a cost. It can inflate false hope or deepen stigma. Science's virtue is not certainty, but correction. The strongest claims about human transgenerational trauma await the most stringent tests. And yet, there is wonder here that doesn't rely on hype. Consider the placenta as an instrument that composes a child's first symphony from the parent's physiology and the environment's key. Consider the idea that a night of poor sleep leaves a faint smudge on metabolic genes measured the next morning, and that months of better sleep can lighten it. Consider a father's decision to stop smoking before conception, altering small RNAs in his sperm. A mother's walk with a friend lowering cortisol during pregnancy. A community's decision to plant trees cooling a neighborhood and quieting stress. None of these are magic. They are mundane, which is their power. They render biological inheritance, not as a verdict, but as a conversation between past and present. Let's return to the house and its photographs. The people in them endured things we are lucky not to face. Some of their echoes may ride in your cells. Many more ride in your family's stories, habits, and hometowns. The science of epigenetics says both matter and both can be changed. It says genes are scripts, not fates. Marks are notes, not chains. It says an echo is not a command. It can fade, be answered, be harmonized. We end then with the mystery where it belongs, not in slogans, but in the open questions that drive good work. How often, and through which molecular routes, do human experiences breach the reprogramming barrier? Which tissues carry the most consequential marks? And when are they most open to change? How do policy and parenting, therapy, and community show up in the methylome and in the life of a child? What counts as repair, and who gets access to it? These are scientific questions and civic ones, braided together like the strands in a helix. So here's my invitation to you, and the question I hope you'll wrestle with in the comments. If the body truly keeps a living score, responsive to danger and to care, what would it look like to build a society that tunes the next generation, not just away from trauma's echo, but toward resilience's song?